My name is Kelly Thorvalson, and from the South Carolina Aquarium, and I'm the program leader for the AZA, that's Association of Zoos and Aquariums Sea Turtle SAFE program. SAFE is an acronym for Saving Animals from Extinction. There are more than 40 SAFE programs within AZA working to protect some of the most endangered animals around the globe. With 18 AZA Zoo and Aquarium program partners, the Sea Turtle Safe program is helping to conserve an amazing group of animals that we all know and love, sea turtles. This is our third live expert webinar, and we're excited to be planning 2 more in 2024, one on World Sea Turtle Day, and we're excited to be planning another in the fall. All of our webinars are live and live on the Sea Turtle Safe YouTube channel. So we encourage you all to watch those past webinars and certainly future webinars that are to come. We're excited to hear from Dr. Brian Shamblin today, but first we're going to take a few minutes to convey some housekeeping information. There are three additional SAFE steering committee members on this webinar: Cassandra Davis from Aquarium of the Pacific, Katie Emmons from Dallas Zoo, and Bridget Cervero from the Maritime Aquarium at Norwalk. They are going to be helping ensure that we have a smooth experience today. This time I'll turn it over to Cassandra to go over housekeeping.
Hello, everyone. As Kelly mentioned, I'm Cassandra from the Aquarium of the Pacific and part of the Safe Sea Turtle Communications team. And just a reminder, today this webinar is being recorded. As Kelly mentioned, it will be uploaded to the Sea Turtle Safe Program's YouTube channel, and it will be distributed to everyone who registered. So please share this with friends and colleagues. We also have the opportunity for you to ask questions live during the webinar. Please use the Q&A feature. You'll find a little Q&A button down at the bottom of your screen, and you can click on that in order to ask questions during the presentation. We'll get to as many of those questions as time allows at the end of our presentation. And participants are able to see all questions. You're also able to upvote a question if it's something that you are particularly interested in hearing answered. This will help us choose which questions to ask if there are more questions than we have time to answer live. The chat has been disabled to avoid confusion with the Q&A. However, we will also be adding helpful links to the chat during this presentation. And with that, I'm going to turn this back over to Kelly to get us started.
I was muted, sorry. All right, at this time, I'd like to introduce our speaker. Dr. Brian Shamblin is an associate research scientist at University of Georgia. His Master of Science and PhD research addressed the genetic structure and spatial ecology of Northwest Atlantic loggerhead turtles and green turtles. The Shamblin Lab uses genetic and genomic tools to inform management of conservation of threatened and endangered species. We're thrilled to have Dr. Shamblin with us today to talk about his ongoing research, to which many of you attending this webinar today contribute to. Take it away, Brian.
Thank you, Kelly. And I want to extend a huge thanks to the entire program for extending the invitation. Before I get started in telling everybody about our genetic tagging project, I wanted to take a minute to acknowledge that this has been a team effort. So we're building on decades of prior monitoring and research, and we couldn't possibly have pulled a project like this off without tremendous support from the state agencies, as well as the sea turtle nest monitoring networks in the southeastern US. So we have a lot of folks who are state, municipal, or federal employees, but we also have a lot of folks who are community science volunteers. So literally hundreds of volunteers have contributed to this research on the nesting beach. And the same holds true in the lab. You know, this is not a one-man show here. We've had over 60 students, technicians, and lab managers now who have contributed to this research going back over the last 15 years or so. So it's kind of hard to distill 15 years worth of data into one talk. I'm going to try to not get too far ahead of myself here. The central theme is going to be spatial ecology. So we're going to look at that from several different angles, but I just wanted to provide you an outline to give you an idea of where we're headed. I'm going to start out with a little bit of background about loggerhead turtle natural history and how the project got started. And then I'm really going to focus on spatial aspects of interesting things that we found. Some of those were anticipated, some of those were really big surprises. So I'm going to talk about nest site fidelity, meaning where a female is nesting relative to where she last nested. I'm going to talk about that at individual and population levels. I'm going to talk about females nesting at what we call the leading edge of the nesting distribution. So those northernmost females up in North Carolina and Virginia. I'm going to circle back to natal site fidelity, which is where a female is nesting relative to where we think she hatched. And then finally, because we so focus on females when it comes to sea turtles, I'm going to try to give the males a little bit of love in terms of what we've learned looking at relatedness of our nesting females about the males. So if we zoom really far out at a global scale, we have several distinct nesting aggregations for loggerhead turtles around the world. These are called distinct population segments. They are designated under the Endangered Species Act based on 3 criteria: that they are discrete, they are significant to the species, and recognizing these allows us to deal with differing threats in different regions, and then recognize that some of these populations are maybe in more dire straits than others. So we're really fortunate in the southeastern US to host the largest of all the global nesting aggregations. Sea turtles have complex life cycles, so there are lots of threats. The oceanic juveniles spend the first several years of their lives in habitat that looks like this, which also means they're potentially interacting with longline fisheries, as well as anything else that may be floating in these sargassum convergence zones. So that could be oil, and more recently, we are increasingly concerned about plastic debris that these little turtles are ingesting. It doesn't get a whole lot safer when the turtles get closer to home. So historically, one of the really big challenges that we think led to declines in loggerhead turtle nesting and why they are threatened here in the Southeast is drowning in shrimp trawl nets. And so there's been a lot of progress on that front with the implementation and enforcement of these turtle excluder devices that have allowed a lot of these turtles to escape over the years. It's probably contributed to what we are now seeing as recovery in some of these populations, but we're still well below historical nest numbers. This is some of Don Witherington's great artwork that was commissioned by the Sea Turtle Conservancy, and it really does convey just how complex these life cycles can be. Genetics have helped play a pretty big role in filling in a lot of these gaps and building this picture. I mean, this looks like a fairly simple schematic, but it truly is the result of decades of research by dozens, if not hundreds, of scientists around the world. And the one thing to note here is that the nesting females are the most accessible life stage. That's the one time where we know those turtles are going to have to come back to land, because those eggs need dry sand to properly develop and incubate. So we often use sea turtle nests as an index or a proxy for overall nesting female population size. So it's much easier to go out in the morning and count nests than it is to actually see those nesting females. For most of the species, that's happening at night. The challenge is when we have data from nesting, we don't have the underlying demographics that are involved. So these are some index nesting beach data from Florida going back to the late 1980s. Things were looking pretty good through the 90s, nest numbers going up, but all of a sudden we see a pivot in the late 90s, and then the numbers start to crash. So, of course, when that was happening real time, for us north of Florida, those numbers bottomed out in 2004. For Florida, they bottomed out in 2007. But of course, in 2007, we didn't know that we had hit rock bottom. We were all extremely concerned this was going the wrong direction. And without additional information, there's really no way to know what's happening. And one of the first questions was, when we have fewer nests, do we truly have fewer females nesting? Because female loggerheads are capable of laying up to 8 sets of eggs in a nesting season, it could be that there are other things happening that are causing these females to lay fewer sets of eggs and potentially stay out away from the nesting beach for longer in between nesting seasons. So the only way to really get those additional information is to have individual nesting histories. And traditionally, those were collected on flipper tagging beaches, still are. Unfortunately, those are logistically challenging. These females, if they emerge at high tide, are capable of coming in, nesting, and getting back to the water within an hour. So it means you have to cover the beach every hour, all night, from May through the end of July. And also permitting is a challenge. So at this point, for the Northern Recovery Unit of loggerheads, which are all the females nesting on the Atlantic coast north of Florida, we only have 4 tagging beaches left, 2 in Georgia and 2 in North Carolina. These represent about 5% of the nests for this subpopulation north of Florida. And these are critically important projects. So these are eyes on these turtles. They're the front line in looking for emerging pathogens, injuries, seeing body condition for these turtles. But when it comes to demographics, the challenge is a lot of these females are nesting on more than one beach. So if you look at historical data from Wausau Island, going all the way back to the beginning of Critter Research Project in 1973, only about 14% of the females that were tagged there returned there in subsequent years. Now, those numbers have gotten a lot better. Some of this was related to tag loss. Some of this was much higher mortality rates back then. But still, the challenge is we know we're only getting glimpses of what's happening on these nesting beaches. And so we're missing what's going on in that much larger matrix of available habitat where nobody's out there at night. So we really wanted to figure out, is there a way that we could capture that information without the need of physically intercepting these females? And we stumbled upon this during my master's work in 2005. We were working with the Little Cumberland Island Turtle Project. At that point, they were still out at night tagging. Dr. Richardson started that project in the early 1960s, and they had a female that came in and nested. Her nest was depredated by raccoons, and then she had made it to the water before they got to her. So the technician had the foresight to freeze some of those eggshells and ask if we could tell if it was one of their tagged females that they were collecting skin samples from. At that point, there wasn't a lot of hope that I could do that based on what I was seeing in the literature. But after playing with it for a few weeks, we gave it a shot and it worked. And we did indeed match that fingerprint to one of their tagged females. So that's when the light bulb went off. We started a pilot study in 2006. And then ultimately, the project grew from there. So the protocol for any of these beaches north of Florida is if you call it a nest, you actually have to dig down and verify that there are eggs present. And so that means that collecting samples for this project it's not too onerous for the network to do it if they're already out there checking on these nests. So we take a single egg from each nest, we chop a portion of that eggshell, and then we ultimately extract DNA from that is reflecting the mother turtle's DNA. So it's as if we have sampled that female directly, except we can do it without the need to ever interfere with her or see her. The fact that we can do it the next morning really opened up the possibilities for doing this on a much bigger scale than would ever be logistically or financially feasible in terms of the direct night work. And we use microsatellite markers to match nest to either known females that we have sampled on these tagging beaches or to each other that then we can create fingerprints from that get added to our genetics database. So what you're looking at here are raw data from a single female. We use 16 markers currently in 3 different reactions. So those markers are those peaks that you see in the different colors there. These are exactly the same kind of markers that we're still using in human forensics. So this is a little bit old school. These days, there's much more modern technology to use, but we still use it for the same reason human forensics hasn't fully switched over to more modern technology, which is we have a massive database now that we would lose all of this information for, and we'd have to go back and re-genotype all of these samples with new markers. We have really good power with these markers to identify individuals. We actually have more power with these 16 markers than the core 13 loci that the FBI originally used for the human forensics panel. So I always say if it's good enough to put somebody in prison, then it's good enough for us to say, yes, this is the same mom, or no, this is a different mom. So this gives you an idea of the study area, and I'm sorry for the Northeast Florida folks that I didn't extend this map south. We started statewide in Georgia in 2008, and we were ultimately able to bring our partner states on board to the north in 2010. So that includes South Carolina, North Carolina, and we even get a small number of loggerhead nests in Virginia, Maryland, and the occasional one north of there, like in New Jersey a couple years ago. In 2016, we were able to get permission to dip our toes over the border and sample on a limited basis in the 3 northernmost counties. So I'll talk a little bit about some interesting findings from there a little bit later. We have ultimately, up through last year, we still have a long way to go for those assignments from last year, but we've assigned 120,000 nests now to 14,000 unique females. So it's been a pretty huge effort to try to collect saturation sampling data for the entire subpopulation. And what we found initially was not too surprising. These are observed clutch frequency data from Wausau Island in Georgia in 2010. So these are based solely on seeing the female at the tagging beach. And the numbers that you see across the x-axis are the number of nests per female. And so what you see is most females detected only once. This is a really common pattern for marine turtle tagging projects globally. And typically we think the reason for this is site fidelity. These females are using more than one beach to nest. And sure enough, if we zoom out regionally using our genetic tagging, we see something that looks much more like a bell-shaped curve. And these turtles are laying on average about 4.5 nests per season. So of the females that nested on Wausau in 2010, 64% of them ended up nesting on other beaches that year. And that creates a lot of challenges for us when we're trying to understand reproductive output, survival, things like that, where we need to see these females to know that they're back. Interestingly, what we found over the years with this project is most females have what we would consider, moderate or strong fidelity to particular nesting regions. So 73% of all the females lay all of their eggs on less than 20 kilometers of beach. But that still often means they're using more than one project beach or more than one island for nesting. And so again, the challenge is if you don't see a female, you don't know if she's nesting right next door or if she may have been lost to the breeding population. And these are just a further extrapolation of those Wausau data. This is a paper that was led by Coretta Research Project, where the black bar on the right is showing all the nests on Wausau that were laid by the females that nested there from 2010 to 2017. But then all these other gray and white bars are other nests by that same group of females on other beaches. And you see most of those are nearby. They're on other Georgia beaches or just across the border in South Carolina, particularly on Hilton Head. But really, we have connections all the way up to North Carolina. So we have a tremendous amount of variation in terms of individual variation in nest site fidelity. And this is just an example of that. This is one particular female's nests. So this red box is highlighting her nesting footprint, if you will, from the beginning of the project in 2009 all the way up through her last detection in 2021. So she's nested as far south as Sapelo Island in Georgia, as far north as Lighthouse Island in South Carolina. So 23 nests, 15 different project beaches. and covering over 250 kilometers of nesting beach. Now, the flip side of that are females that have really strong site fidelity. So this is the same base map. If you zoom in and really squint north of the photo there, north of Savannah, you'll see a little blue box beside Hilton Head, and that is indicating Pritchard's Island and Fripp Island. So this is Mama Pritchard. She was found in really bad condition trying to nest on Pritchard's Island in 2008 by the volunteers. She was taken to the South Carolina Aquarium for rehab, ultimately released in the spring of 2010. And you see here Kelly at the release, and also Dr. Shane Boylan. He gave me a blood sample for this turtle, and we were able to enter her in our genetics database. Unfortunately, 2010 was a year that we did not have coverage on Pritchard's Island, so she probably nested, but we didn't detect it. She did come back, though, in 2013. And so since 2013, she has now been responsible for 31 nests over this really small area of 6 kilometers of beach. And really, most of those nests are on about 3 kilometers on the north end of Pritchard's Island. So when it comes to site fidelity, the variation doesn't just occur at the individual turtle level. We also are consistently seeing variation across the entire nesting range for this subpopulation. So loggerhead turtles use the Earth's magnetic field to navigate at large spatial scales. And those magnetic isoclines are hitting the coastline basically like lines of latitude. So what we did in this case was we used these lines that you see here to break that study area up into these latitudinal bends to get treat them as a rough proxy for latitude. The white bars that you see to the right of that are showing how far apart on average the females' nests are in each of those bins. So if you look here, kind of in the middle, the strongest nest site fidelity, so where the females are spreading their effort over the smallest area, is in bin 6, which happens to be where Cape Romaine National Wildlife Refuge is. This is the highest nesting density for loggerhead turtles north of Florida and accounts for about 25% of all the nests for the entire subpopulation. Those nests are on Cape Island that runs north to south and Lighthouse Island that runs east to west. And these are some historical imagery that show you how the islands have changed through time, going all the way back to the 1984 here. If you kind of fast forward 20 years, you don't really see a huge difference here. And even all the way up to 2011, you don't see a whole lot of change happening here. At that point, Cape Island accounted for 27% of all the nests in South Carolina, and Lighthouse Island accounted for 6% of all the nests in South Carolina. Well, as some of you may have remembered, if you've been involved in sea turtles going back to this period, at the end of the nesting season in 2011, we had Hurricane Irene blow through in August, and that little skinny part of Cape Island, kind of right in the middle, got blown out. What was a washover fan got totally breached and turned into an inlet that has since never healed. And so what we've seen is this really slow transition of some of these turtles that historically probably nested on Cape Island moving down to Lighthouse. So that really in the last handful of years, Lighthouse has just as high a nesting density, if not higher, than Cape. The reason all of this is concerning to us is on the heels of Hurricane Idalia last year, both Cape and Lighthouse were essentially completely overwashed. And so it was very late in the season. Most of the nests had already hatched by then. But these are some photos of the west end of Lighthouse Island taken by Michelle Pate, the South Carolina DNR Sea Turtle Coordinator. You see all the rack built up on the back side of the island. There's really not a whole lot of nesting habitat out there. And unfortunately, it still sounds like it looks like this. There's just not a lot of accretion that has happened. And so it's a really big concern for us that the highest density nesting region for these turtles in this subpopulation, it's where they have the highest nest site fidelity. These females know this is where they want to be, also has really limited real estate. And so it leaves managers of the refuge and of South Carolina DNR Sea Turtle Program really asking questions about, how do you deal with this situation? Clutch relocation is one potential option. We try to avoid that where we can to leave the nests as natural as possible. But even if you are okay with relocating nests, in this instance, there's so little dry sand available that there's really not a lot of places to move them to. And this potentially could be what we're seeing in the future with sea level rise and just changes in dynamics at these barrier islands. So this is kind of a window into what we might be expecting in the future in other areas. And I'll circle back to this Cape Romaine situation a little bit later. While I'm talking about site fidelity, the other thing that I wanted to highlight is notice that the turtles nesting up in North Carolina in general have weaker site fidelity than those further south. There's lots of potential reasons for this. I think part of it is based on the shape of the coast. If you notice that we have these three large crescent arcs that happen between each of the capes, I think that potentially obscures nesting cues that the females are using to select their nesting beaches. What's interesting, though, is we noticed really early in the project that in central and northern North Carolina, the females were marching to the beat of their own drummer. So it's not just that they have weaker site fidelity. There's many aspects of their life cycle that seem to be different than other parts of the subpopulation. The proportions that you see here in red for each of these bins are the percentage of females that we identified in 2010 that we never saw come back over the life of the project. And fortunately, these numbers got a lot better starting in 2013. But the spatial pattern that you see here has stuck consistently every year, which means for these northern nesting areas, we're much more likely to see these females nest in only one season and never see them again. And we're much more likely to see them only laying one clutch of eggs. And so if we were seeing those patterns at one beach, we would say, well, maybe they're nesting right next door. But in this case, we're looking right next door and they're not there. And so we really were scratching our heads with this pattern. Where could we possibly be missing all these nests that should be there for these females? And we pretty quickly dismissed the idea that it was based on survey effort, because these beaches are getting covered just like any others for the project. One of the things that we considered, but we also dismissed early on was, well, wait a minute, there's plenty of loggerhead nesting down in Florida, but all the previous research, my research for my master's thesis suggested that those should be a different subpopulation of turtles. So the pie charts that you see here are showing us the relationships of mitochondrial DNA haplotypes. This is a maternally inherited DNA, comes from the mom down to the offspring. And essentially each color here represents a different female family lineage. So there's not a lot of diversity. Really, most of the females are either an A1.1 in blue or an A2.1 in yellow, but note how those wedges in those pie charts change as you move from north to south. The Northern Recovery Unit north of Florida, all the individuals are in blue. By the time you get down to Central Florida, you start seeing a lot more of those turtles in yellow, down at Cape Canaveral, down at Archie Carr National Wildlife Refuge. Of course, the challenge is we do think our interpretation here is if these females are hatching within these central eastern Florida beaches, then they're going to return somewhere in that region to nest. But because we don't have a lot of diversity with this marker, we can't really pick up turtles that are moving across these boundaries. And so small numbers of turtles that are kind of breaching that boundary between Central Florida and Northern Recovery Unit, we would never be able to detect with this mitochondrial marker. As I was digging through the literature, I found a note in Herpetological Review from 1981 where a turtle tagged at Cape Lookout in North Carolina was detected by Doc Earhart nesting at Cape Canaveral 19 days later. And so I think the story was already out at this point, over 40 years ago. But I think a lot of this was dismissed as, well, it's just aberrant behavior. It's not very important. Not a lot of the turtles are doing it. You have to kind of consider the context here, though. Only 30 females were tagged at Cape Lookout that year. So the fact that any of them were detected in Florida was sort of a hint that there might be something bigger going on. And sure enough, when we look at females, when we started sampling in Florida from 2016 up through 2021, we've actually found over 60 now that have nested both in North Carolina and in Florida. About half of those did that in the same year. About half of it did it across years. And this is what a couple of those ended up looking like. So this is a little note that we published in Herpetological Review, sort of a 40-year revisit. This turtle started out nesting down near St. Augustine in Florida. 21 days later, she's nesting up at Cape Lookout. So typically we would see this happen more like every 2 weeks. So it took her longer to do it, but 3 weeks later, she's nesting in North Carolina. And then about 3 weeks later, she's all the way back down in Florida. And no sooner had the ink dried on this paper than I was looking at some data from 2019 when we found a female that could even beat this. So she nested all the way up on the Virginia-North Carolina border. 21 days later, she's nesting down in Florida. And then 17 days later, she's back in North Carolina. So these females, I totally get hedging your bets, not putting all your eggs in one basket. This seems a little bit extreme to me. From an energetic standpoint, this is pretty wild. It's costly to move around that much. So we don't really know why they're doing it. But we do think the fact that we have females straying at this scale is really valuable. This is an insurance policy for these populations. These are the females that are going to be colonizing new nesting sites. And so we always thought that the further north you went, the younger those populations were, the more recently they had been colonized. But I think we all thought that that was in the rearview mirror. You know, that's something that happened hundreds, maybe even a few thousand years ago. I think what we're seeing with these data is that this colonization is happening in real time. Like, we are watching this, we are documenting this process, which I also think is valuable because it's showing us what the potential is for these turtles to adapt to climate change and potentially colonize new nesting areas. So this turtle ended up swimming nearly 1,500 kilometers at minimum to be able to put those nests in those 3 spots. To me, the really valuable aspects that we can gain from the genetic tagging come from the fact that we can use the same markers that we use for individual identity to say something about relatedness. This is just another example of those raw data. In this case, we have 2 different females. So marker 28 on the left is showing that this marker doesn't match. We only have one of the 2 variants there matching. But then on marker 73 in green, we see that we do have a match. So both variants for these 2 individual females are matching. So these are 2 distinct females, we can say that, but the pattern of matching is also telling us that these turtles are sisters. And so it's really valuable for us when we can connect those generations through time. And one of the things that I did recently was look at recruitment. So looking at those new first-time nesters in the population. And for the group of turtles that started nesting between 2016 and 2021, there were about 5,000 of those. We were able to assign 1,300 to a known mom in the database. What you're looking at here is complicated, but we have moms on the left. They're assigned to those latitudinal bins based on the central tendency of their nesting throughout their entire known data. And then on the right, we have their daughters, their assigned daughters, and where they're nesting. And so if we look at those Cape Romaine moms, we see that the vast majority of their offspring are nesting right there at Cape Romaine or very nearby. But we also see arrows fanning out across the entire subpopulation. So in this case, the thickness of those arrows is conveying the magnitude of the connections between these sites. So most of these offspring tend to be right where mom is in Cape Romaine. And ultimately, Cape Romaine is responsible for 40%. Moms nesting at Cape Romaine can account for 40% of all of those new daughters that we could assign to a mom. So it's not just that Cape Romaine is super important from a nesting density and a nest numbers perspective, but also from a recruitment perspective. And if you think about this from the perspective of those receiving bins, so if we think about it from the daughter's perspective, you see that that tiny little arrow that went from bin 6 to bin 8 in the previous slide all of a sudden is really thick now. And so in this particular case, from the South Carolina border up past Cape Fear, there actually are more new daughters nesting there that have mothers nesting down at Cape Romaine than have mothers nesting on these same beaches. So to me, this just reinforces how critically important Cape Romaine is for the overall subpopulation. And, we like to draw these boundaries, but the turtles have zero respect for these state lines. And this is an example of a family that actually illustrates this connection. So what we have here is mom's nesting site on Cape Island as the blue star. And then she has 7 daughters that have recruited into the breeding population. And the colors that you see for each of those daughters correspond to different dads. So in this case, we actually have 2 pairs of full sisters. And in each case, we have a situation where we have one sister nesting in North Carolina and one sister nesting down near mom, near or in Cape Romaine National Wildlife Refuge. So, of course, this begs the question, why? How is this possible? Why do we have sisters nesting so far apart? Could this be that we have really weak natal site fidelity in this part of the nesting range? Well, one of the other things that we discovered is an idea that's been around for a really long time, but now that we've been doing this project for so long, we can actually document this process. And that is, we're seeing shifts in nest site fidelity behavior through time with nesting experience. So here's a family from Georgia primarily. So this is also really reinforces the value of the partnership that we have with the physical tagging beaches. Because of all the work that they did in previous decades, we have a case here where we have a mom boomer that was physically tagged by Coretta Research Project in 1988, but she now has a genetic tag also associated with that physical tag. So we know she nested up through 2012. Well, she has 3 daughters that have recruited into the breeding population. And we've been able to follow those now through time to see how their fidelity has changed. Her first daughter, actually, we only have one nest for her, which is fairly common in a lot of these first-time moms. So we can't really say anything about how far she's spreading her nest because we only have one nest that year. When she came back in 2019, though, she initially nested up on Kiowa Island in South Carolina. And then she's like, nope, this is the wrong spot. Came back closer to home on Wausau and Ossabaw. And when she came back in 2022, she's using only 4 kilometers of nesting beach. Her second daughter nested as far south as Cumberland Island and as far north as Blackbeard Island in her first nesting season. When she came back in 2019, she's nesting on Little St. Simons and Sea Island, so that nesting range has shrunk down to 13 kilometers. And then when she returned in 2022, she's using only 2 kilometers of that nesting beach. And then finally, this third female initially crawled on Jekyll Island, nested as far north as Blackbeard. And when she returned 4 years later, that nesting range shrunk from 54 kilometers to 7 kilometers. So we truly think these females are using that first nesting experience, at least in those early years of coming back. It's been 25, 30, 35 years since those turtles were home. And we think they're using that first nesting year as sort of on the job training. They're recalibrating their navigational cues in that internal GPS unit during those initial nesting years. So that finally brings us to the males. There's a lot we don't know. Most of the work that has been done by Dr. Jake Lasala that leads Moat Marine Lab's Sea Turtle Research Program. The data that we have for the NRU was generated in collaboration with the Coretta Research Project when Jake was a master's student at Georgia Southern. He found a lot of multiple paternity, so females mating with multiple males. Interestingly, in 3 years of sampling, never found the same male across different females' nests and never found the same male repeated through time. And so To me, an important takeaway of this is when we look at our relatedness data and we see full sisters, so same mom and same dad, that likely means they hatched in the same year. That's our assumption, at least. Well, all of a sudden, if we can say that these turtles are hatching in the same year, then we start to get little bits of insight into what survival may look like for these hatchlings from hatchling stage up through adulthood. And so the majority of these turtles we cannot assign to a known mom because their mothers stopped nesting before the genetics project started. A much higher proportion of these turtles can be assigned as potential siblings to one another. And so the biggest shock of any of the analyses that I've done recently for our last final report was of those 5,000 new kind of first-time moms from 2016 to 2021, 1 in 5 of them, 20%, had another sister in that cohort. So if you stop and think about what I just said about the fact that probably means they hatched the same year, you do a little bit of math, say we're generous, 6 nests, 115 eggs per nest, assume we have 100% emergent success, which pretty much never happens. Assume these are all females, also probably not. We're talking about a maximum of maybe something on the order of 700 potential female sisters hitting the water. Also, remember what I just said about multiple paternity. So all of these things are going to draw this number down, but best case scenario, we're talking about 700 or so potential sisters hitting the water from one mom in a potential nesting season. The fact that we're seeing such a huge percentage of the population with sisters suggests the fact that we may have really strong differences in fitness across these moms. So a lot of you probably have heard or seen, 1 in 1,000 reach sexual maturity, 1 in 3,000, 1 in 5,000, 1 in 10,000. Understandably, it's very hard to estimate those numbers. And we're not saying that this says survival is better than we thought it was, but what we are saying is either survival is better than we thought it was, or we have some moms that are doing way better than replacing themselves, whereas we have other moms who potentially could nest for 30 years and never have a daughter make it back to the nesting beach. There's lots of potential reasons for this. Could be maternal investment, could be where these females are putting their nests, could be something inherent genetic or epigenetic involved. I don't have the answers, but I think we need to ask those questions and try to get some of those answers. So circling back to this idea of no repeat males, that either means there's a lot more males than we thought there were out there right now, or it means that there's maybe a lot of mating occurring along migratory corridors, or that males are moving around. And these are some data that were recently published from Dr. Simona Ciriani and colleagues using stable isotope analyses to figure out where these turtles are living when they're away from the nesting beach. So there are 4 major foraging areas for these turtles that are color-coded there. Most of our turtles nesting in this part of the Atlantic coast end up going north at the end of the nesting season. So they're foraging in the mid-Atlantic bight in purple. But also note that there are chunks of purple in those pie charts for the turtles nesting down in central Florida as well as southeastern Florida. So there's lots of opportunities for gene flow to be happening at really big spatial scales here. And sure enough, we see evidence of that. So if we look at relatedness of our new moms, so now we're not looking at hatchlings, we're looking at relatedness of these new moms, these first-time moms. We have situations like this where we have this mother-daughter pair down south in blue. So we have mom and daughter nesting really close to one another. And then up north in North Carolina, we have mom and daughter nesting really close to one another in purple, Onslow Beach. These 2 new daughters were assigned to the same dad. So even though we have these females nesting 400 kilometers apart, we have the same male potentially contributing to both. So it does suggest that we have mating along migratory corridors and or males are moving around during courtship season. which basically tells us, just like with the females, we have to be very careful about how we interpret data from any one beach. This is something that we really need to be looking at across really large regional scales. I just wanted to close by saying that overall, the nest numbers look really good for this subpopulation. We're definitely seeing encouraging signs of recovery. We're seeing over the course of the project roughly a 7% annual increase in annual female nest numbers, or sorry, annual female numbers that we're inferring from the nests. That's fantastic. That's really good growth rate. We're not sure how much longer that's going to occur before things start to stabilize. But all that being said, with sea turtles, everything happens slowly. So we're really happy that things are headed in a positive direction. But I would say that we're not necessarily out of the woods yet. We have a lot of emerging threats, and we also still have a lot of unanswered questions. So all of this work that we've done has been to feed a loggerhead population model that the managers can use to plug those threats in and see how they may impact population recovery. And we still have a lot of data gaps in trying to inform those models. This is a project that has survived. For many years, we were funded through grants. The last 2 years, we are surviving on donations. So we've definitely changed how we've done things. We're trying to be as absolutely efficient as we possibly can be. But one of the things that we may have to do moving forward is scale back sampling. The goal was always to try to collect a sample from every single nest. That may just not be possible moving forward. So one of the things we're trying to do now is explore different subsampling options to figure out how we can get the most bang for the buck. We can stretch those donation dollars as far as we possibly can. And one of the ideas that we're contemplating is actually starting to try to sample more dead hatchlings during nest evaluations. So some of the really big remaining questions are, how old are these turtles when they reach sexual maturity? How long can they nest once they do? Right now, we get all that information from skeletal chronology work. The challenge with that is the turtles have to be dead to be aged, and we don't really know what's killing them in a lot of cases. So a lot of them are being artificially removed from the population before their time really came. And so if we have other ways to get that information, we can try to capture what is possible, at least for these females, if we could mitigate some of these threats. And that's where we're really excited about a lot of markers that are kind of coming online, or at least we think are possible in the near term. Part of this would be epigenetic, so not the genes themselves, but the switches that turn them on and off. There are possibilities there for both aging, estimating chronological age in these turtles, and also sexing hatchlings. That would really open up a lot of possibilities. The other thing that's concerning, because sea turtles have environmental sex determination, is not only are we worried about population feminization, so hot chicks, cool dudes, the hotter, drier nests are going to produce more females. At some point, that might be an issue, but also tends to produce hatchlings that are smaller and maybe can't crawl as fast, maybe can't right themselves as well, swim as fast, potentially makes them more vulnerable to predators. So there's lots of this kind of work happening. I would love to see us integrate this for our Northern Recovery Unit subpopulation. We have a really rich long-term data set, and I think it would be a shame to not try to build on that and capture as much of this type of information as we can. So I'll just close by saying, if anybody is interested, a lot of the volunteers that are part of the monitoring network are on Facebook. So we do have a Facebook page that we try to update fairly regularly. And then I have a QR code here that will take you to my lab website, where there's information, more information about the project in general. And also, if anybody is interested in donating, we do have a 5013C University of Georgia Foundation account where we can receive donations that can go toward this project. So thanks for having me, and I'll be happy to take questions.
Brian, thank you. What an amazing presentation and just fascinating research. I loved seeing Mama Pritchard in there. She was the largest adult female loggerhead we had ever treated at the South Carolina Aquarium. I think she was close to 390 when she was released. And she was in our facility for 2 years with massive boat strike wounds, and in her carapace, and so it's just really cool to see an animal like that continue to thrive in the wild, and that you can track her contributions to the population through this research. It's really amazing. So we have 4 questions. You guys, please put your questions in Q&A. I'm going to turn it over to Cassandra and Katie to field questions for Brian.
Thank you so much, Kelly. And Brian, that was so exciting and so wonderful to hear about the research you're doing. And I think it sparked a lot of really interesting thoughts and questions. We're going to start out on one that I think started off very early on, and I think it could help. You answered a little bit of it, but let's see if we can dive in further. Kathy asks, why are there so few tagging beaches left?
I think a lot of it just comes down to expenses. It's pretty logistically challenging. On the developed barrier islands, you have to deal with people that are on the beach at night. On the undeveloped barrier islands, we often have erosional areas called boneyards, where we have erosion into the maritime forest. That makes it really tricky to navigate in the dark. So between just the logistics, the expenses involved, and all the permitting that's required to be out there, a lot of folks, I think, just decided that it wasn't worth the effort anymore and that they wanted to put that effort elsewhere.
Our next question is from an anonymous attendee. I'm wondering, could the females be nesting in multiple different places to increase the likelihood of survival for their hatchlings?
So we think that's probably related to how natal homing started, right? So these moms have no idea if their nests are going to survive or not. And that means these new females really have no idea where they should be putting their nests either. And if that's the case, then where do you put your nests? You put them where you hatched because you hatched there and it worked. And so we think that's why we have natal homing in the first place. But obviously that would be detrimental. If all the turtles went back to the beach where they hatched and something bad happens to that beach, then that means bye-bye population. But if some proportion of these females are constantly sort of moving around and testing out other areas, then it's that insurance policy that allows the population to persist. So I'm not sure how much conscious thought goes into it, but certainly those females that are moving around are helping ensure that these populations are making it.
That's really interesting. And I think that some of the talk on females sparked some thoughts about the males as well. So Terry asked, does the data on males indicate that males only procreate once in their lives since they don't show up in multiple years or multiple females?
I don't think so. I think it just shows the limitation of only looking at this from a single beach perspective. Really, the only way to rebuild these male fingerprints is to sample a lot of their hatchlings and then compare that to mom. And so that's logistically pretty challenging. You know, you have to watch these nests carefully and sample the hatchlings as soon as they emerge. So understandably, that's limited the data that we have on the males to just a handful of beaches. But I think what our data is showing likely is happening is that those males are mating with females across really large areas, or they're sort of passing each other as ships in the night, where we have a male that is sitting on a courtship site, and we have a female that's coming from her foraging area toward the nesting beach. And so they're passing when the female is receptive, and that's allowing the opportunity for gene flow to be happening at these really big spatial scales. So I think it's more of how we're looking than what's actually happening. I think it means that we need to look at larger regions.
There were a couple questions that addressed fundraising. They wanted to know what fundraising efforts are being done to continue the research, and if we can drop the link again in the chat for where they can donate.
I am open to suggestions. A lot of the fundraising is happening at the grassroots level, at the individual projects. I'm open to anything and everything that we can do to try to make this, more sustainable, but this is not my area of expertise. But if there's anything I can do that would help, I'm certainly game, because I think it's critical that we continue to do this. It may not look like it always has, but I think continuing it at some scale is going to be really important. And, one area that we haven't really tapped into, I think, are some larger foundations, private foundations that might potentially be interested. But again, that's sort of out of my realm. So any help that anybody wants to give us in that regard would be greatly appreciated.
Hey guys, so we are at 1:59, and although questions keep rolling in, I don't know, Brian, we didn't talk about if you have 5 minutes beyond 1 o'clock to stay for Q&A, but for those that have to jump off, I'd like to just give some closing remarks, and then maybe those that want to stay on can do that. So I just want to first, sorry, let me get back to just want to take give you a huge thanks, Dr. Shamblin, for joining us today and for all the great work that you and your team are doing to advance sea turtle conservation and this really fascinating research. I want to thank Sandra, Katie, and Bridget for helping keep us organized, because you wouldn't want to be part of this webinar with just me. I definitely want to thank, and I'm just going to share my screen really quickly. Want to thank our 18 program partners for the Sea Turtle Safe program and all of the support they provide. These are some amazing institutions that are doing great work around protection of sea turtles within their walls and beyond their walls. And so just kudos to everyone here. And then ultimately, thank you for all joining us today. Whether you are supporting sea turtles, supporting conservation as a whole, volunteering on nesting beaches, funding sea turtle work, however you are supporting the environment and sea turtles, we are grateful for you and for you joining today. If you have any questions about Sea Turtle Safe, please email the email that you see there, safeseaturtle@gmail.com. Thanks everyone, and have a great day. If you'd like to stay on for a few minutes, we'll stay and answer questions. Thanks everyone. Oh, did I? Looks like I didn't actually share. My apologies, you guys. Here are our program partners, really amazing institutions working to protect sea turtles. And there is our email address. All right, and I will stop share. And we'll get back to some questions. They keep rolling in.
Absolutely. And I wanted to highlight, there were a few questions about this, and I'll admit I'm on the West Coast and I don't know much about it, but we had quite a few people wondering about gathering DNA from tracks rather than from an egg. Would you expand on that?
Yes, Dr. Duffy's lab down at the Whitney lab is leading the charge on that front. The challenge is you need enough DNA to identify individuals, because that's what we're trying to do here. You certainly can get mitochondrial DNA. You can get bits of nuclear DNA that way, and we are collaborating with Dr. Duffy on that front. We've had, so far, we've only tested 24 samples. We've had one success in those 24 where we actually got a full maternal fingerprint from mom that matched the egg from that nest. But that also means we had 23 failures or partial failures. So back at the very beginning of the project, we tried external swabs of the eggs where we're trying to get epithelial cells that were shed by those moms. We think a lot of the DNA is actually trapped within the calcium matrix of the egg. So unfortunately, that means, destructive sampling of that one single egg. But we are continuing to explore that swab technique and with eDNA basically in the sand to hopefully try to get to the point that we are not taking an egg at all.
There's also a couple of questions regarding how beach nourishment or other restoration projects might be affecting the nests or the natal homing of the sea turtles?
That's a good question. Unfortunately, we don't have a great amount of baseline data there, and we don't have a ton of renourishment happening in the NRU compared to what is typical in Florida. It seems like, though, these mamas know where they want to be, and they're there regardless of conditions. So it's kind of similar to what we're seeing in Cape Romaine. They're going to try to nest there, even if the habitat is suboptimal. At some point, if they false crawl enough, if they're hitting a seawall or they're hitting a scarp enough, they may decide to ultimately move, but it's not their first choice. They really know where they want to be, and that's where they go.
Similar to those questions, there were a few asking about the changes in water temperatures and weather, storms possibly affecting where they lay their nest, the cue to come into nest, and questions about how global warming affects the number of female hatchlings, and environmental change affecting the habitats of the nesting beaches.
Yeah, lots of questions, not a lot of answers on those fronts. The challenge with the hatchling sex ratios historically was you had to either sacrifice the hatchlings or you had to bring them into captivity to grow them large enough to do laparoscopic work safely. So the beauty of these potential epigenetic markers is that we could take a tiny skin sample or blood sample potentially to get that answer, which really opens up doing the sampling on a much larger scale. And then also pairing that sampling where we're watching these nests with data loggers. So we know moisture content, we know temperatures. I think that'll be really valuable moving forward. In terms of how females are going to respond, I'm just kind of nervous based on what the females at Cape Romaine are doing, because there certainly is sand not that far away. And there's a little bit of nesting on the adjacent beaches, Raccoon Key, Murphy Island, Cedar Island to the north. But again, those turtles know where they want to be. And I think as long as there's any dry sand there at all, they're going to try to nest there. And so it leaves us kind of asking, how do we deal with that? Do we just let it play out? It's sort of a classic ecological trap at this point. And so, do we intervene and how do we intervene really at this stage? There's only so many places that we could safely move nests to, even if we do decide to relocate at large scale. So I think it's important that we try to at least track the process, because it really could be what we can expect maybe in other areas moving forward.
Just following up on that one, in general, are there things that any of us can do to help sea turtle nesting habitats? What would you recommend?
I just think getting involved locally is important. If you can volunteer, if you live near a group that does any of these surveys, I think trying to help with local lighting ordinances, development, trying to preserve as much habitat as possible is like really critical at this stage, I think. And I think a lot of that needs to happen locally where it has the greatest potential to actually see change.
Here's a quick question. Can you gather DNA sampling from an egg that was destroyed by a predator, but the shell remains?
Yes, and we do that. So any nests that have been depredated or where an egg is accidentally broken or maybe even crushed by the female, we use that. We don't take a viable egg. The challenge at the end of incubation is the maternal DNA that's present in any undeveloped eggs essentially has rotted for 2 months in really hot, humid conditions. So we're sometimes able to get those to work, but usually we get just partial fingerprints from those. And the other challenge when we're trying to get a sample from every single nest is storms and things like that. So if we don't get that sample the morning after, then we may lose it altogether.
And I think we have time for one more question. And a couple of them have been about predation. So continuing on what you were just talking about, has there been any increase in predation, any efforts to prevent predation? What are you seeing on the nests right now?
That's kind of out of my realm of expertise, other than kind of seeing the data as they come in. I don't think there's any huge spike in predation. I know the biggest issues really have been with coyotes in a lot of areas where they're kind of showing up and they're figuring it out. They're really smart, and also feral hogs can be super destructive. And particularly when the sows figure it out and they have piglets, then those piglets pick up on it fairly quickly, and so they can be really destructive to sea turtle nests. But I don't think, other than those two issues, I don't think predation is necessarily any worse than it ever was. And most of the species that are involved are probably natural predators, maybe subsidized by us, like raccoons, eating trash or things like that near the beach, but probably things that always ate sea turtle eggs and are part of the natural system.
Hey, Brian, is there a website that, this was a question, is there a website we can go to look at the different females and where they lay their nests? Like the type of data you've just presented, is that somewhere on the website? I know you do post updates on the Facebook page, so maybe that would be where folks should go.
The general data are in, well, like summary data are in seaturtle.org, and I can send that link afterward. It's basically seaturtle.org/nestdb/genetics. And that's an overall summary of the project. And there are pull down menus there for year, for state, and for individual beaches if you want to go check the summary data out for those.
Nice. Fantastic. Well, I think we'll leave it there. You guys, thanks everybody for joining us, and thank you all, and mostly thanks to you, Brian, for being here today and this amazing presentation and for all the work you're doing to further sea turtle conservation.
Thanks for having me.
Have a great one, guys.